Hello and welcome. In this lab session, we will learn how could you predict the protein 3D model or a structure by using the threading algorithm. As we already discussed previously that for the prediction of protein structures, we have different algorithms. But so far, ITSR is considered as more efficient program due to its high accuracy prediction. At the end of this lab session, you will definitely have an idea about how could you predict the 3D model by using ITSR, which is an online server. So let's have an overview of ITSR. ITSR is such a hierarchical approach that is used to predict the protein 3D structure and the structure-based function annotation. How ITSR predict the protein 3D structure? Firstly, it identifies the structural templates from the PDB by multiple threading approach, LOMITs, and then drive by rethreading the 3D models through protein function database. So in this way, ITSR predicts the 3D structure and the structure-based function of the target sequence as well. So let's have a look on some practical example. To get into the website, you just have to click on the above mentioned link. You will be redirected to their main home page. In the home page, as you can see that we have in the menu bar different options and you can get the basic information about the ITSR, that how could it predict the protein structure and functions of the proteins from their home page. The use of ITSR is super duper easy. You just have to put your sequence in this box. But firstly, it is important that you have to use some credentials. You have to use your commercial email ID, and before that you must have your password as well. For that you just click here and firstly register yourself. After registration, on the mentioned email ID, a password will be sent to you. By using such email ID or password, you can easily use your ITSR server and you can easily use your account. So you have to paste your query sequence, which is your amino acid sequences over there. Your sequence must not exceed the 15,000 residues of amino acids. And after that, you just have to click on Run ITSR. A message will appear that your job ID number is this, and you will be notified by email once your job is completed if an email has been provided. आपकी mentioned email ID पे आपको मैसेज आ जाएगा when your structure has been predicted and it will almost take 1 or 2 days. So these are the results of your query sequence which you have submitted. As you can see in your result page, firstly this is your query sequence which you have submitted in the FASTA format. It also predicts the secondary structure of your query sequence, like which part of your amino acid is going to represent the helices and which part of your amino acid is going to represent coils and strands. The confidence score represents that if you find the confidence score near to 9, it means like we have some of the confidence score that is mentioned over here is 9, some of them is 4, 5, 7. If your confidence score is near to 9, it means that your prediction accuracy is high. But if it is near to 1, then it means that your prediction accuracy is low according to the server. In predicted solvent accessibility, in this portion, you have an idea about the solvent accessibility that you will it will let you know that which amino acid will be present at the surface of your protein, while which amino acids are buried inside the core of the protein. As you guys may know that amino acids that are hydrophobic means that are non-polar, that are not water loving, these are buried inside the core, while the hydrophilic are present at the surface. So at this point, you can have an idea about the localization of your amino acids in your protein structure. Under the heading of predicted normalized B factor, आप फाइंड आउट कर सकते हैं which of your amino acid residues at which positions are more flexible and which amino acid residues are non-flexible. So as in this graph, you can see that these amino acids which are above this line, above this zero line, these represents the amino acid residues which are more flexible as compared to these which are present at the under this line or under this zero bar. Under the heading of top 10 threading templates used by ITSR, the templates in this section are the 10 best templates selected from the LOMID's threading program. Usually one template of the highest Z-score is selected from the threading program. So in this case, as you can see that we have different templates mentioned over here along with a different Z-score. Z-score is important. If it is above 1, then it means this template has good alignment with your query sequence. So जितना highest Z-score होगा और near to 1 होगा, that template have a best alignment with our query sequence. Usually, ITSR predicted top 5 models, but sometimes it can show 2 or sometimes it can show 3 models as well. So the confidence of each mod is qualitatively measured by C score. इस मॉडल की क्वालिटी को मेजर किया जाता है ऑन द बेसिस ऑफ देयर सी स्कोर. अब सी एज यू कैन सी ओवर देयर कि हर मॉडल के अंडर आपको RMST वैल्यू नजर आएगी, आपको सी स्कोर नजर आएगा. तो मॉडल की क्वालिटी को मेजर किया जाता है थ्रू देयर सी स्कोर. अब सी स्कोर है क्या? सी स्कोर इज टिपिकली इन द रेंज ऑफ -2 and 2, where a C score of a higher value signifies a model. जितनी higher value C score की होगी among all the models, उस मॉडल की significance means उस मॉडल की quality as compared to the other models बेहतर होगी. So as you can see over there कि हर एक मॉडल के अंदर आपको C score मिलेगा. तो आपने हर एक के C score को एनालाइज करना है, देखना है कि कौन सा C score है वो हमारे पास हाई है. Then you can use this model or download this model for your further research. यहाँ पे हमारे पास डाउनलोड की ऑप्शन भी है. When you click on the download model 1, तो ये मॉडल्स PDB फॉर्मेट में डाउनलोड हो जाएंगे in your local disk, जिसको आप किसी भी प्रोटीन विजुअलाइजेशन टूल में इन प्रोटीन के स्ट्रक्चर्स को फर्दर एनालाइज कर सकते हैं. There is another feature in the ITSR that due to the structural similarity, these proteins often have a similar function to the target protein. Previously हमने डिस्कस किया कि ITSR on the basis of their structure functions को भी आइडेंटिफाई कर सकता है. लेट्स जस्ट से कि आपके पास जो आपका क्यूरी सीक्वेंस है और आपने जो मॉडल को प्रिडिक्ट किया है, यू डोंट हैव एनी आइडिया अबाउट देयर फंक्शंस. आपको उस प्रोटीन के फंक्शंस के बारे में नहीं पता. तो ITSR विद द हेल्प ऑफ द TM अलाइन क्या करता है, PDB में जाता है और ऑन द बेसिस ऑफ देयर स्ट्रक्चर अलाइनमेंट परफॉर्म करता है. और जिन स्ट्रक्चर्स की अलाइनमेंट हमारे क्यूरी हमारे टारगेट सीक्वेंस के साथ और हमारे प्रिडिक्टेड स्ट्रक्चर के साथ जिसकी क्वालिटी अच्छी आती है, उसका TM स्कोर हाई होता है. तो अकॉर्डिंग टू TM स्कोर वी कैन प्रिडिक्ट दैट कि the PDB structure with this PDB hit have good alignment और similarity with our predicted target. तो हम ये कह सकते हैं कि इस प्रोटीन स्ट्रक्चर की सिमिलैरिटी फंक्शन के अकॉर्डिंग हमारी टारगेट प्रोटीन के साथ हो सकती है. So in this way you can find out the functions of your predicted protein structure as well, and you can model your 3D structure of your protein by using the ITSR. Thank you.